No escucho. Lo siento, pero no escucho. Sorry? I cannot hear.
Good morning. Good morning, Ms. Candidate. Hereby I open this academic ceremony in which Ms. Caterina Giovagnoni will defend her academic thesis entitled Lipids and Lipid Transporters, Key Role in Membrane Dynamics, Inflammation, and Alzheimer's Disease. May I invite you to present in the next 15 minutes a summary of your studies and the major conclusions of your thesis. I'd like to give you the word.
Thank you. Dear Prorector, member of the Corona promotion team, colleagues, family, and friends. In the next 15 minutes, I will talk about my PhD project entitled Lipids and Lipid Transporters, Key Role in Membrane Dynamics, Inflammation, and Alzheimer's Disease. In this presentation, I will go through three main topics. What is Alzheimer's disease, the relation between lipids transporters and Alzheimer's disease pathology, epigenetic changes in Alzheimer's disease. Alzheimer's disease is a neurodegenerative disorder that affects cognitive functions, behavioral abilities, and interpersonal relations. It's the sixth most common cause of death in the Western countries and is negatively affecting families, communities, and healthcare systems around the world. There are no treatments that can arrest the development of this disease. The pathology of Alzheimer's disease is characterized by events happening in the brain that can lead to the neurodegenerative process. We have, in fact, a protein accumulation in the frontal part, mainly in the frontal part of the brain, a widespread inflammation, lipids and lipid transporter disbalance, and epigenetic changes. All these four events are singular events that together can influence each other and can bring to the neurodegenerative process and therefore to Alzheimer's disease. In the next slide, I will mainly talk about lipids and lipid transporter disbalance and the relation with Alzheimer's disease. The main cells of the brain are called neurons, and as most of the cells in the human body are made by a membrane constituted by lipids. Lipids are fatty molecules highly present in the brain and can be divided in different subclasses according to the biological and the biochemical properties. And one of these subclasses are sphingolipids. Also, sphingolipids are highly abundant in the brain and can be divided in two main categories. The ceramide, which is the smallest sphingolipid, and the slingomyelin, which is a more complex sphingolipid that derives from the ceramide. Both ceramide and slingomyelin are important because they are building blocks of the cell membrane, like they are like bricks of a wall. But they are also very important because are taking part to the formation of the so-called vesicles. These vesicles are part of the membrane that detach from one cells and attach to another cells. Sphingolipids are very important for the formation of these vesicles, but also for the messages that these vesicles brings along. The first person that described the pathology of Alzheimer's disease was the German doctor Alois Alzheimer, who described already in 1906 an aberrant lipid metabolism when he examined the first brain of the first Alzheimer's disease patient, August D. Since then, many scientists try to understand the relation between the lipid metabolism and the pathology of Alzheimer's disease. And now, after many researches, we know that patients who suffer from Alzheimer's disease have low concentration of sphingomyelin and high concentration of ceramide in the brain, meaning that there is a disbalance on the concentration of sphingolipids in the brain of people suffering from Alzheimer's disease. But the question is, what is causing this disbalance of sphingolipids in patients of Alzheimer's disease? So let's imagine the cell as a house with different rooms and different compartments. We know that the ceramide binds to a protein that is called CERT, ceramide transfer protein, in a room of or in a compartment of the cell. And the CERT is able to transfer this ceramide from one compartment of the cells to another compartment of the cells. In this compartment, the ceramide undergoes two biological changes, biochemical changes, and the sphingomyelin is formed. The sphingomyelin, as I said before, is very important to have a well-structured cell membrane because it's part of the cell membrane, but it's also very important to the buildup of these vesicles and the messages that they bring along. But it can happen that CERT doesn't work and doesn't bind to ceramide. So ceramide is moved to the wrong compartment of the cell. And therefore, it doesn't undergo to any biochemical changes, and the sphingomyelin is not produced. We end up with a not well-structured cell membrane and also dysfunction of messages and vesicles. This phenomena also have a consequence because we think that there is also an increase in concentration of ceramide inside the cell. So our working models are the following. If CERTs work and binds to ceramide and can transfer ceramide from one compartment of the cells to the other, there is a balance of the sphingolipid production, meaning that this could lead to a well-structured cell membrane and also a good communication in between cells through these vesicles. These might lead to a very well-functioning brain. But if CERT doesn't work, we think it might be the cause of the disbalance of sphingolipid, therefore to a more concentration of ceramide and less concentration of sphingomyelin, which we think might lead to a dysfunction of the cell membrane, also dysfunction of communication between cells. This, we think, might be the cause of the neurodegenerative process and therefore of Alzheimer's disease. So the aim of my study was to unravel the role of CERTs in influencing the building blocks of the cell membrane and the messages between cells, understand the role of CERTs in inflammation, and study genetic variation of sphingolipid networks in Alzheimer's disease. In Chapter 2 and Chapter 3, we wanted to investigate whether CERT was in the cell membrane. So we label CERT in the cell, in the outer part of the cell. And as you can see here from the picture, we demonstrated that CERT is part of the cell membrane. You can see we label CERT in red and the cell membrane in green, and therefore when we see the yellow signal means that the CERT is actually part of the cell membrane. Afterwards, we wanted to investigate whether CERT not only is in the cell membrane of the cells, but also influence the messages and the formation of these vesicles. Therefore, we increased the number of CERTs inside the cells, and we actually saw that when we increased the number of CERTs inside the cells, we also had an increase in communication and increase in formation of the extracellular vesicles. So as you can see from this picture, we have in blue a cell, and in red we stain the cell membrane. In yellow, you can see that the CERT is localized in the cell membrane. And from this graph, you can see that when the cells were treated with CERTs, we have an increase in number of vesicles, therefore increase of communication between cells. In Chapter 4, we wanted to investigate whether CERT was involved in inflammatory processes. So we demonstrated that CERT was binding to a protein that is called STAT6, a protein that is well known to be involved in inflammatory processes. As you can see here from the picture here, we stain a cell brain, a brain cell where it's always in blue, and we stain STAT6 in green and CERT in red. So in the yellow means that CERT and STAT6 binds together and colocalize in the cell. Afterwards, we wanted to investigate whether this binding, so the colocalization and the binding between SERTs and STAT6 was having some consequences on the sphingolipid composition of the cells. Therefore, and we actually demonstrated that the binding lead to a less concentrated ceramide and inside the cells, which, coming back to our working models, could be seen as a good sign. So in fact, as you can see here from this graph, when the cells were treated with STAT6 and SERTs, the concentration of the ceramides were decreasing inside the cells. In the second part of my presentation, I will mainly talk about the epigenetic changes and the link between epigenetic changes and lipids and lipid transporter disbalance in Alzheimer's disease. So let's imagine the genetic, I will mainly talk about the genetic information. So let's imagine to have a building plan. The building plan, a good building plan, brings to a well-structured house with good wall and well-structured wall. But besides a good building plan, we also need a good material to have a good house. Same happens with the genetic information. When we have the genetic information, we have healthy brain via or thanks to also the transcription and the expression of the genetic information. But it can happen that the building plan is not okay, or the material that are used to build up the house are not great. And we end up with a broken house and not well-structured cell wall. Same happens with the genetic information. If there is an insult or a change on the genetic information, we also have and we might have also a change in the transcription and in the expression of this genetic information, which we might end up with a neurodegenerative process and therefore to Alzheimer's disease. So in chapter 7, we wanted to investigate whether there were some genetic variation or epigenetic changes in genes connected to sphingolipids and the differences between the genes related to sphingolipid in Alzheimer's disease patients' brain and healthy control. So when we did a comparison of genes connected to sphingolipid in healthy brains and in brains from patients suffering from Alzheimer's disease, we demonstrated to have 103 genetic variation in Alzheimer's disease patients related to sphingolipids, meaning that there is 103 genetic variation in Alzheimer's disease patients compared to healthy control. Afterwards, we did some mathematical analysis to show if there were some candidate genetic variation able to revert the Alzheimer's disease phenotype. And we actually saw a set of genetic alteration that if perturbated, could lead to or could revert the disease phenotype towards a more healthy phenotype. This set of genetic alteration could be seen as a potential target for further studies. So in conclusion, my PhD studies brought 3 main key points. SERT can be used to monitor cell membrane, can be used as a potential target to change the number of vesicles, concentration, and composition. SERTs are able to bind STAT6 and together can reduce the concentration of ceramide in the cells. And the expression of sphingolipids related genes is different in the brain of people suffering from Alzheimer's disease. These results open up new research opportunities and have identified possible actionable biological targets to counteract Alzheimer's disease. And I thank you all for your attention, and I give you the word back to the prorector.
Thank you very much, Ms. Candidate, for your clear presentation, well illustrated and also well in time. So congratulations. We're going now to discuss, and we have 6 opponents. 3 of the opponents are online, and 3 of the opponents are here present in the aula. And I would like to start with the first opponent, which is Professor De Baas. He was also the chairman of your Assessment Committee, and he is affiliated as Emeritus Professor of Chronic Inflammation and Neurological Diseases at this university. And I'd like to give him the first word.
Thank you, Mr. Pro-Rector. Dear candidate, congratulations with your beautiful thesis. It's a very comprehensive work. I will also compliment the promotion committee and also especially compliment for the layout of the cover, which usually is done by other people. You did it yourself. Congratulations. Of course, we are not here to congratulate you all the time. We're going to ask you some questions. And my first question would be, as you demonstrated, Alzheimer's disease is a multifactorial disease involving many, many proteins. Why did you use CERT? I mean, an answer to study in relation to this disease. One answer could be because my supervisor discovered that protein, but I'm not going to take that as a correct answer. You please answer my question.
Highly esteemed opponent, thanks for your question and your nice words. It's correct, it's true. So, Alzheimer's disease is a multifactorial disease with many proteins involved in the pathology and the pathophysiology. I decided with my promotion team as well, we decided to study CERT because of the importance of the sphingolipids in the pathophysiology of this disorder. And I'm relating CERT to the sphingolipids because, of course, it has been discovered that CERT is the only protein able to transfer the ceramide from the ER to the Golgi. So, and is also essential to process this, the ceramide to build up the sphingomyelin. And since it has been shown many times that people suffering from Alzheimer's disease has a disbalance of sphingolipid composition inside the brain and inside the also the blood, studying CERT would be, for us, would have been the most straightforward way to understand how this dysregulation is happening. Moreover, it has been demonstrated that CERT binds to a protein that is called SAP, serum amyloid protein, which is also important in the pathophysiology and the pathology of Alzheimer's disease. Therefore, seeing all this consideration and also since CERT has a very important properties and functions also during neurodevelopment, in the maintenance of the neurons, and in general of a healthy brain, we wanted to investigate whether this CERT was dysregulated or which was the role of CERT in Alzheimer's disease.
Okay, thank you very much for your answer. My second question would be more specifically related to chapter 4, and you clearly demonstrated in figure 3 on page 98, which you also showed the slide, is nice co-localizations of the two proteins CERT and STAT6.
Yes.
And in the next figure, on figure 4, you showed, and you also showed in the presentation, that STAT4 is able to reduce the ceramide content and also other proteins, which is to be expected, of course. But my question would be, how would you take that further in your research project to show relation between STAT6 and neuroinflammation? Because one of the functions also of CERT is that it's activating complement, it's binding to SAP. So how would you take that further if you will have, you know, a couple of €100,000 and say you can write a project? How would you take that further? What sort of experiments would you like to design?
So I've been thinking a lot about the experiment that I would perform to prove that this binding actually have a meaning, biological meaning in inflammation. Of course, I would be, at the beginning, I would demonstrate again that they actually co-localize with a more, I would say, fine experiment. It's called PLA, proximity ligation assay, thanks to which, if two proteins are together or very close, we have a fluorescent signal, which is kind of different of the than the two proteins co-localize in the cells through this through this assay. Then I would go more for a functional assay testing inflammation. So I would maybe I would use immune cells of the brain to understand if this binding actually is bringing or is beneficial to
What are the immune cells of the brain?
the microglia.
Okay.
I would study the cells in microglia. And to see maybe study the cytokines, the chemokines to understand if this binding or decolocalization of this protein inside the cells is leading to a less inflammatory pathway. Of course, we can do to study this. We can do through several assays like FACS analysis. We can also study with qPCR, so the gene expression. We can also study through protein expression or by ELISA. Nowadays, there are a lot of assays that with few samples, we have a big readout of data, which would help us to understand in which direction this or pro-inflammatory or anti-inflammatory this binding can drive the cells. And since in our lab we just now developed new animal models that is a knockout of SERT, I would use also the knockout of SERT in microglia. So I would use this model to see whether the function of STAT6 that is well known to drive the cells in an anti-inflammatory phenotype is through SERT itself. So knocking down a gene and studying the.
Okay, so assume now you spend all your money, and so how? And the final question, if the director allows me, no, it doesn't allow me anymore. So I'll give the word back to the prorector. Thank you very much.
I'm very sorry, Professor Dwaas, but 7 minutes are over. We have to proceed, and the opposition will be continued by Professor Honing. Professor Honing was also a member of your assessment committee, and he's affiliated as professor in analytics in systems imaging at our university. And I'd like to give him the word.
Thank you, Mr. Prorector. Dear candidate, I've been reading this with a lot of joy, looking to a lot of biology and biological systems. But before going into, let's say, the content, I have some philosophical questions about the propositions. And may I ask one of you paranames to read proposition number 5?
The body does what it wants. I am not my body. I am my mind.
Yeah, that's nicely said by Rita Levi, but why did you choose for this proposition, and how does this fit in your thesis?
Highly esteemed opponent, thanks for your question and your nice words. So first of all, I wanted to acknowledge an Italian neurologist, Nobel Prize, that for me was also a leading figure for my career. So there is a personal reason for that, why I put a quotation of her in my thesis. How does it fit in my thesis? Well, I studied the brain, and the brain is in close relation with the mind. I wouldn't say they overlap, but they kind of overlap. So I also, with this proposition, I also wanted to acknowledge the importance of the knowledge, and the importance of the knowledge of the brain.
But would you agree? Because I completely disagree with Rita Levi.
Okay.
I was taught there is a triangle between mind and body, and actually there's a lot of psychosomatic diseases where the mind decides something, and then the mind does whatever it wants, and then the body has to follow. So still, how do you see this?
I do agree that that there are several diseases that have a close relation between mind or brain and body. I didn't put this proposition thinking about diseases. I put this proposition thinking more about the knowledge and the knowledge about the brain. But if we think, nevertheless, I acknowledge your comments because it's true. Maybe we cannot agree totally with Rita Levi, but I do still think that the mind has a more powerful, has more power than the body.
I can follow. I think I follow your thought, because if you look to Alzheimer, the body or the biology is doing something, and you as a mind, as a person, have to follow this degeneration of let's say the biology. And that's maybe last question, because you used also the word Alzheimer disease. I would say, is disease a real good word for this? And are we not just speaking about aging or degeneration and not really a disease?
Yes. So I would call it disease. So it might be. So that's thanks a lot, because this is a very good point, actually. I think we should split up aging, so neurodegeneration related to aging and neurodegeneration related to Alzheimer's disease. Alzheimer's disease has some pathological hallmarks that the normal aging, the normal brain aging doesn't have. So I would say that yes, it's an age-related disorder. Most of the cases are age-related disorders, but we also know that 1% of the Alzheimer's disease cases are also early onset. So
I think I agree, because as a more molecular scientist, I was looking to your disbalance between ceramide and the Slingomyelins, and actually for sure that has something to do with the inhibition or the improper performance of the enzyme taking care of this chemical transformation. So there's definitely something in the transcription of the DNA to the protein mistake. So you could say it's a disease. I have still one question, I think, Mr. Prorector, and then go to page, let me see. Yeah, I have it here, 51. And I'm looking much more always to molecules and not so cellular levels. And in figure 2.8G, you showed some structures, high resolution, medium resolution, but you explained that you were using AlphaFold 2. And although AlphaFold 2 is I would say, a paradigm shift in science, it is only, and I would say only, a artificial intelligence-based thinking of what the molecule could be, and there's no modeling, no theoretical modeling. So why did you use this approach to correlate molecular structure to biological, or let's say more phenomenological effects, and not traditional X-ray, NMR, or even molecular modeling, because there's a lot of knowledge on molecular modeling of these not too big proteins?
Yes. So there is, CERT, to the best of my knowledge, CERT was not crystallized, so we don't know exactly how the structure is. It was just crystallized the pH domain, so the initial part of the protein and the end part of the protein, but the whole protein was not crystallized. So it's true that these are models, so it might not confront the reality, it might not be the reality, but we thought that since this model nowadays are very close to the reality, what the structure is in reality is, we thought they could be interesting to at least understand a bit how these two proteins could be interacting and could be also structured while they were interacting.
Perfect. Thank you. Thank you, thank you for your questions. Hans.
Thank you very much. Then the opposition will be continued now for the 3 next oppositions online, and I would like to welcome Dr. Ledesma. She's affiliated as group leader of the Department of Molecular Neurobiology at the Centro de Biology Severo Ochoa in Madrid, and I would like to give her the word.
Okay, thank you very much, and good morning, everyone. Congratulations, Caterina, for the very clear presentation and for the nice work. And I extend my compliments to your mentors and your team. So my first question is more technical. One of the merits of your work is the development of a technique that will see raft lipid dynamics, the use of a SERT antibody together with the cholera toxin. So my question is, what are the benefits of this technique in your opinion, with respect to others that are already existing? Do you think this is a global way of visualizing rafts, or is more a sphingolipid related technique? And if so, do you think that changing or have you tried to change other lipid composition in rafts that are not sphingolipids, and then what would happen to your probe?
Esteemed opponent, thanks for your questions and your nice words. I would like to repeat the question because there was some sound problems. So
okay.
You're asking me what is the benefit for us to use an antibody-based technique to visualize the lipid rafts, as I explained in chapter 3 of my thesis. And what is the advantage of using such a technique over the already existing technique to study lipid drafts? Is that correct? Because I think I'm
correct.
Lost some.
Yeah.
Yes. Okay. So we wanted. The purpose of this chapter was, first of all, to understand if SERT was in the lipid draft. The lipid draft are microdomains with many sphingolipids, and it was also shown that besides many sphingolipids, there is also a high concentration of ceramide. So we wanted to investigate if SERT was also binding ceramide in these microdomains. This technique would be useful because most of the probes of the antibodies that are binding to sphingolipids are technically quite difficult to use. So we thought that maybe using antibodies targeting proteins in this case would be, let's say, not better solution, but a proof that actually the probes that are binding, or the antibodies that are binding to lipids, are actually working. Lipid drafts are quite complicated microdomains in the cell membrane to study because they are resistant to detergent. So sometimes it's quite complicated to study the composition of these microdomains. Therefore, we thought, yes, that there are many techniques that are used to stain and to deattach or to understand the composition of this microdomain, but use an antibody-based method would be more, let's say, workable also technically, rather than other techniques that are more complex, more long, and maybe also time-consuming.
Okay, thanks. So a second question goes more to your epigenetic studies and the transcriptomic studies. So this has a great load of bioinformatic analysis, even mathematic analysis, as you mentioned. So I have two questions referring to what was your involvement in this kind of analysis, bioinformatic and mathematic? And the other is from I think another merit of your work is that it opens so many possibilities now. So from these genes that you have picked up, would you choose one or two, and how would you analyze them in the context of Alzheimer's? If you would choose one of the list that you have came out with, what kind of experiments would you do?
I'm very sorry, but I didn't hear the first question.
Is the involvement in the bioinformatic and mathematic analysis on your epigenetic studies, and then which of the genes that you discovered there, choose maybe one, and just elaborate why you would choose that and how you would test its participation in Alzheimer's.
So the epigenetic studies we did on the single lipids related genes was a study to understand if there were some epigenetic regulation on Alzheimer's disease patients that were related to single lipids. This is because single lipids are, as I also illustrated during my presentation and all my thesis, are gaining ground on the Alzheimer's disease field because of their role in the pathology. So we did, in fact, discover many genes that were upregulated or downregulated or with epigenetic changes in Alzheimer's disease patient. And yes, I think I would pick few of them to study further. For example, the CAV1. CAV1 is a gene that is the protein that is encoded by these genes, the calveola 1, is very important because it's a part of the lipid raft. So I also come back to my previous chapter. But it's also very important because it has been shown that is dysregulating in Alzheimer's disease. So is a good candidate to study further with genetic models or with some biological assay or some functional assay to further study this the association of CAV1 with Alzheimer's disease. I think this one would be my first, let's say, the gene that I would study as first. But we discovered many genes that are very important in the pathway of single lipids, the pathway of the single lipids are involved too. So I would start with CAV1, but I would go for sure with others in relation not only to Alzheimer's disease, but also in the dysregulation in the single lipid pathway.
Okay, so I don't know if there's time, but just the third question is you were partially answered before. It's about the.
I'm sorry. I am.
Okay.
Dr. Letzmer, I'm sorry to interrupt.
I'm very sorry,
your time is over, so we
No
have
problem.
to continue. I'm sorry.
That's okay.
I didn't hear very well. I'm very sorry I didn't hear very well the first question.
No problem.
We continue with the next member of the assessment committee, which is Professor Rusticelli. He is professor in physics at the Istituto Nazionale Biostrutture e Biosistemi in Roma, and I'd like to give him the word. Professor Rusticelli.
Yes, thank you. First of all, I would like to congratulate Caterina for following in excellent way the strategy of Galileo Galilei, who said, I'm looking for the light of science and its benefit. So in your case, understanding biological phenomena and looking to clinical research application. And now, because time is precious, I will combine my question in a single one so that you can distribute the time for answering. So the first general question is if you can continue this work, and in this case, what are the results that you hope to obtain, especially in the direction of clinical application, or if not you, if somebody else will continue, could you answer anyhow this question? And then second, within this question, a sub-question is related to the first, your answer to the first question that was addressed to you this morning. That is, you answered that you will consider the use of animal models. My question is, if you know the technology of organoids, and if you think that by producing organoids, that by using the technique of Nobel Prize Yamanaka, you can obtain also from human men, from skin, an organoid which simulates the brain. You can have a brain with pathology and another healthy, and compare and see the influence of drugs. And then the other part is if you think that in addition to all your techniques, you could use also small angle neutron scattering to get information within the inner information of extracellular vesicle. And so the last one is a specific, what kind of information could you...
Professor Rusticelli, I would like to give the student opportunity to answer first these two questions, and then we continue.
Okay. Thank you.
Can you answer the first two?
Yes.
Okay.
Highly esteemed opponent, thanks for your question and your nice word. Maybe I repeat the first question. You're asking me if someone else or myself would continue the work, what I would do, and also with the use of animal models, if I'm correct. Is that correct? Yes. So in relation to my work, I for sure will continue study the ceramide and the sphingolipids, or the sphingomyelin modulation in the cells and in the brain. I will go more in details in my statement. So I would study a bit more in detail how modulation of sphingolipids could lead to pro-inflammatory or anti-inflammatory processes. We know already, it has been shown already, that the sphingolipids, the different species of sphingolipid have different roles in inflammation and towards a more pro-inflammatory or anti-inflammatory phenotype of, for example, immune cells. But still, there are some knowledge gap that I would feel if I will continue working on that. So that would be my let's say first aim. Regarding the animal models, I didn't have the chance to work a lot with animal models. I did work for some paper with animal models. I do really think that they are very important to understand some biological pathways and some biological changes that are happening in diseases or in specific conditions. So for sure, I would also use animal models to study my aim and to continue my studies. And the second question was, could you please repeat the second question? The organoids. Organoids. Yes. So yes, an alternative of animal models. I think it's a quite groundbreaking technique, the development of organoids. Specifically, I think it's very important to use the use of organoids to study neuron and brain-related diseases, because it makes let's say easier to develop some, for example, drug studies or some more functional assays without the use of animal models. So I think is the use of organoids are getting ground more often in the last period. So I think it's quite useful technique to use and to study and to develop further. Not only, as I said, for drugs, but also for drug development, but also for other type of studies, epigenetic studies or.
Okay, thank you very much, Caterina.
Very much. Then the opposition will be continued by Professor Mitricic. He is affiliated with the Department of the Laboratory for Stem Cells at the Croatian Institute for Brain Research in Zagreb, and I would like to give him the word too.
Hello, everybody. Good morning. My really warmest congratulations, Caterina, for your very good work, and to your mentors and to everybody involved in this great project of your life and important step. So I have several questions, but I will try to simplify them. So again, we already mentioned today a ceramide transfer protein, CERT, which is indeed a very interesting molecule. And in several places in your thesis, you mentioned a link between CERT and cell death, especially of neurons. So if tomorrow you will be a great famous professor and you will teach to your students, what would you tell them? What are possible links between CERT and cell death?
Highly esteemed opponent, thanks for your question and your nice words. I did mention many times indeed CERT and cell death, but I also did mention the role of CERT in contrast to cell death. So here I also need to make a connection with sphingolipids, because as I said, CERT is according to our working model, is very important for the formation of sphingomyelin. We know that ceramide, so the precursor of sphingomyelin, is in relation, or better, provokes cell death. So having an increase of ceramide inside the cells leads to apoptotic signals, leads to cell death. So we see the function of CERT as in contrast with this apoptotic signal, because CERT is helping, let's say, ceramide to get the cells to get rid of ceramide and transform the ceramide into sphingomyelin, which in many cases, I say in many cases because it's not always like this. In many cases, it has anti-apoptotic signal. So, moreover, it has been shown also in a very recent publication that mutation of CERT. So, when CERT doesn't work, or there is a mutation of CERT, we have also dysfunction in the neurodevelopment, meaning that the function of CERT is important also for the neurodevelopment, and therefore to maintain the healthy, let's say, healthy cells. So,
Okay. Yes, thank you. Very good answer. So I will follow based on this first answer, also to link this a little bit with neuroinflammation. You're also talking about neuroinflammation, which is for sure in the background of majority of brain diseases, including Alzheimer. So, for example, you are mentioning also sphingosine as a double sword molecule. Am I right? So what can you tell us about that? Again, there is a link between development and cell death.
That's it. Sphingosine is a metabolite of sphingolipids. So it's formed by a breakdown of ceramide. And yes, has two roles. So can be pro-apoptotic or anti-apoptotic. So can have a role both in the cell death and in let's say cell maintenance. As many sphingolipids many sphingolipids have this role, actually. Many sphingolipids can be seen as pro-apoptotic, anti-apoptotic, can be seen as anti-inflammatory or pro-inflammatory, like for example, the ceramide. Short-chain ceramide are anti-apoptotic, but long-chain ceramide are pro-apoptotic. So according to the biochemical composition of the also the sphingolipids, we can have this dual role and these opposite functions inside the cells. So I think we cannot really generalize saying this lipid is pro-apoptotic or that lipid is anti-apoptotic, but I think we need to really understand the biochemical composition, so the chain length, the double bonds of the lipids, to really understand what is their role in the physiology of the cells.
Thank you very much. This will be, I think, enough from my side. Thank you.
There's room for one more question if you want.
Okay, do we have time? Very briefly, you talk about amyloids, yes, and of course soluble amyloids. And what do you think about? I mean, if you read papers, recent papers from Alzheimer disease pathology, there is more and more mentioned disbalance between soluble and insoluble amyloids. Could we, in theory, link this balance imbalance with sphingolipids or SERT, or this is a little bit more philosophical question and more for the future? So to write your next projects, how would you think? Could you possibly link imbalance between soluble, insoluble amyloids and sphingolipids, for example?
We know that there is a close relation between the formation of fibrils, amyloid fibrils, and ceramide, because ceramide in the lipid drafts are actually stabilizing the enzyme that cuts the APP and then that leads to the formation of fibrils.
Okay.
So yeah, I think it would be very good to understand the relation between the concentration of this ceramide, for example, in the lipid draft or in the cell membrane, more than inside the cells, within the cells, and the formation of these fibrils, which at the first in the cascade, the first part of the cascade are soluble, then they become insoluble. So I think it would be really interesting to understand when also this balance of sphingolipids are happening. Are they happening before the soluble fibrils are formed, or afterwards? Is this a cause or a consequence? I think it's quite a very interesting research question and topic.
Exactly. So it would be interesting to see whether it's cause or consequence. Exactly. I agree with you fully. Thank you very much. Thank you.
Thank you very much. Then the opposition will be continued by Professor Buurman. He is Emeritus Professor of Metabolism and Immunity at our university, and I would like to give him the word too.
Thank you, Mr. Director. Dear candidate, I've read your work with really interest. I was a little bit disappointed you didn't go further into complement, which I hoped in the beginning you would go, but anyhow, it's very nice work you have presented. But now we have to go along and I have to come to some questions. Alzheimer's disease is a multi-headed dragon. You have studied part of the basic mechanisms, but as I said, it's a multi-headed dragon. There are probably many basic mechanisms involved in the onset of the disease. Now, such a disease has proximal mechanisms, and as I said, probably many. There are final or distal mechanisms which are leading to function loss and structure loss. And if you now look into what has been done in the last 20 years, what do you think would be the best to go forward? To look into the structural basic work, or to go more for the distal mechanisms? Because as it looks now, the distal mechanisms are more the same, and the proximal mechanisms are very, very different. The disease has very many different ways. It can be very slowly, it can be very fast. And what would you choose? To go further into depth in how these different mechanisms are, or would you choose for the distal mechanism?
Highly esteemed opponent, thanks for your question and for your nice word. Personally, I would choose more the mechanisms that are driving the pathology. If I understood correctly your question, you're asking me if I would go more to the mechanisms that are bringing to the pathology or the consequence, or yeah, I would study more the mechanisms that are underlying the disease. So I would study this, maybe the structural changes or what is happening inside the cells to better delineate the pathophysiology of Alzheimer's disease.
But if you look at recent data, I think that we have much more possibilities to come forward to look at the more distal mechanisms, like the action of complement, which is playing a role in all disbalances of tissues, and whether it's ischemia, perfusion, even in the head. You will see that if you block the inflammatory reaction, then repair will take place, and you have much less damage, as we see in many other organs. And here we have so many different mechanisms leading to disbalance in the tissue in one way or another, but finally all leading to inflammation, and that inflammation leading to dysfunction, malfunction, structure loss. Yes. And so why would you still choose for the first one and not? I mean, we have going on for so many years, and we have no way of treating these patients, no way at all. We have no future at this moment for treating these patients. Whereas if you see the first results with the complement blocking experiments and complement receptor activating experiments, they're very, very much positive, giving lots of possibility, and that's more the distal. Still, you think you want to? Do you think we can go further when we go to the proximal aspects?
Well, I don't. The thing is that I answer as I would continue my research. I would really try to understand what are the basic mechanisms that are leading to this disease. This doesn't mean that I do not acknowledge also the importance of studying the more, let's say, distal or consequences of these small changes. I think tackling the inflammation, as for example, tackling the C1q is of great importance to at least cover one side, one of the multifactors that are leading to Alzheimer's disease. So I would still trying to study more.
You're right to do so. I mean, you're right to choose. I think even science for science is very well correct to do, because it's the only way we come forward. But I think personally, I think for treating patients in the future, going for the basic for the distal mechanism would be better. But you would choose for science, your own love in science. That's what you be your choice.
Yeah, but this doesn't mean that I do not acknowledge, for example, clinical studies that are that have been done, and they are done nowadays to tackle the distal or the consequences of Alzheimer's disease. But I also do really think that these clinical trials and these are happening also because someone studied the basic mechanisms that are leading to the disease. So I think these two aspects are complementary. So one cannot exist without the other. So yeah, that's what I would do and think.
I think it's very correct. Thank you very much for your answer.
And the beadle gets in exactly on time, so very nice. Dear Miss Candidate, dear Miss Caterina Giovagnoni, a different word. The time appointed for defending your thesis has now passed. The degree committee will now withdraw, not so much to discuss the quality of your thesis, because that has been approved, but we're going to discuss the way you have defended your thesis this morning. I request that you and your company here on the first line await the results of our deliberations and our return to this aula. Thank you very much.
Thank you.
No one stumped you on any questions, so they're not going to have to rehash.
But if did I answer?
Which was fine. Yeah.
And she said, well, I got her fun. So if you then compare it to why would you if they're that lenient which is always the possibility. No one cannot say. But I could have gone to the I drank the oil. But you know, this is really necessary. No. For me, my handshake.
Dear Caterina Giovagnoni, the degree committee here present has assessed the quality of your thesis, but in particular, the way you have defended this thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Martinez is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and that's why I invite your supervisor now to take the floor.
I have to ask you some questions. So, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Caterina Giovagnoni, cum laude, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
There will be a laudatio presented by Professor Martinez, and I give her the word.
For this moment for you. So I would like to tell a little story. So the central nervous system has always been a challenge. As early in 1873, it was very difficult to study because the cells were hard to identify. So the available tissue staining techniques at that moment were not very useful for studying the nervous system. And then there was, at this point, there was a figure in Italy, so that was called Lombardi Camillo Golgi, who set up a simple laboratory on the kitchen of the hospital. And then he started trying techniques on tissue of I think he used the brain of the owl. And then he was able to set up a technique using silver staining that is called the Golgi Method, that is still used in for staining the brain, which depicts the neurons very well with the dendrites. It's a very nice staining. He also identified the Golgi apparatus and also the Purkinje cells in the brain. And I think many people are still using his methodology. So he received a Nobel Prize together with the Spanish scientist called Ramón y Cajal at the beginning of the 20th century. And I think they have the Nobel Prize in Medicine. So I think that later on, so we have antibodies that were developed, and then we can stain very well ion channels and proteins. And in this process, it has been much easier to understand how the brain is structured and how it works. And only in the last 5 to 10 years, the methodology have improved to study lipids. And I think we are at the level of what Golgi discovered so many years ago in respect to lipids. I mean, proteins have been advanced on the study of proteins due to many techniques, but not the study of lipids. And I think this comes to you, come back to you, Caterina. I still remember when you came to Maastricht for the first time from Ancona, which also connects to your story of your PhD. You were an Erasmus student, a very motivated master's student, and you were always focused in your career. I am doing this for my career. It's very important for my career. I do all this because I really have my dream in my career. This word was coming up all the time through the year of as master's student. And to work with CERT was very, very difficult, was a very difficult protein to work with, but you still stick with us and decide to stay for the PhD. So, which indicates that you are not afraid of difficult things and that your ambition for a career and for discovering new things or things that are difficult was there always present. So I think you have done very important work. For example, being able to identify that this protein cell has an important function in vesicle release from cells, and also that these vesicles are important way to communicate so they are very important to communicate between nervous cells besides synapses. So what we thought about synapses being the most important way to communicate between nerve cells is not the case, because vesicles have also an important role. And I think we have to focus on that. You also were identifying many proteins that interact with cells, so making it very easy to understand, or much easier to understand what is the physiological role of this protein. So I think that's all to you, and also how to maybe monitor the dynamics of some lipids by looking to the protein using antibodies. These were all important things you did during your PhD. But I also have to say that this work was not easy, and many times nothing worked at all. So no method was working, and we didn't know what to do. So we were so tired that we went to ask colleagues, maybe with a fresh mind, they will give some feedback, they will give us some ideas what to do. We were completely stuck. And I remember one case of a very knowledgeable colleague that told you, but Caterina, why you don't quit this protein? The solution to your problems is to pick up another protein and to quit this protein, because there is no way you can study it. And then you came to me and you didn't know if this was a joke or reality. You didn't understand if he was really joking, and told me, but Pilar, I cannot quit this protein. I have to go on studying it. So that was your answer to this comment. So I want to say that also you did a lot of things during the Corona time. So the Corona time put a lot of pressure on you because of you had to choose between your career dreams and your family and stay with the people you love, and especially far away. So that was very, very hard in the Corona time. And you fell a little bit down at times, but then you kind of were reborn like the Ave Phoenix. And from there you grew and you were able to finish your PhD and to continue working, and then have again optimism about your future and about your career. So I don't know, I also would like to say that you contributed very much to the pandemic time to make it nicer for the team, because you organized things like we had a cooking experience by Zoom, so everybody of the team was cooking something typical from their country. We learned to prepare gnocchi from you. That was very nice. We ate that several days. That was super delicious. It was, to be honest, the first time I was eating that. And you also organize a sport event, so by Zoom. So we always do high intensity interval training, and we did some yoga, and it was very nice to stay connected in the pandemic. I want to come back to this point that that Golgi will have been quite jealous of the techniques that you have now to study things compared to what he had at that moment. So he will be very proud of you that you have done so many things, and also that you can also not that via the Golgi, you are able to also go beyond with the vesicles and understand what's happening there. So I think that's very nice. And just lastly, I want to say that you should not allow anything or anybody to stop your dreams, and you should always go for following your dreams. And with this, I want to give you my most sincere congratulations in your achievement today, and wish you that you invest this wisely and do something important with it, that the university has, the chance that the university is giving you today. And I would like to compliment the auguri, Caterina, and I would like congratulazioni per il tuo risultato di oggi, and anche congratulazioni alla tua famiglia. I want to thank also Professor Rutten and Dr. Lawson, because it has been important despite supporting the PhD, also the members of the reading committee, the pro-rector, and the members of the Corona today, as well as the Neurin group, because they help also very much in the preparation of the presentation and giving feedback to Caterina to do it well. And I think like this, together as a team, it works very nicely. So thank you to everybody, and congratulations. And for Pilar, thank you.
So I would also like to say, dear Dr. Giovagnoni. Sounds good, I think, yes? Sounds good, yes, thank you. I would also like to congratulate you on behalf of the Board of Deans of our university. In my congratulation, I also put your promotion team, who in the last, I noticed the last 5 years, and I even think 7 years, because you started already in 2015 in the Erasmus program here in Maastricht. I noticed in your file, they brought you to this point this morning, and I would like to congratulate them. I also would like to thank the members of the Corona, also the online members. Thank you for joining us online. It's always much nicer to see you live, that you really actually persons. So I hope that we have a chance in the future maybe to meet here on site in Maastricht. I also like to give my congratulations to the family. I know you from Ascona or from Jerry, I noticed there was the city. And it's very nice that you had the opportunity in this, I would say, post-Corona time. I'm not quite sure to say this, that you came all the way from Italy to visit us today and to be here present, where your daughter family is really has defended her thesis in a very well way, that she got her cum laude. I also would like to mention that the other team members behind there. So I'm looking there, and I was just counting who will be next in line standing here in this committee, but I see somebody gets nervous, somebody not, but okay. I will be here also for the next theses, okay? Dan, I see you already there. Yes, perfect. So by this, I would officially use my hammer. I have a hammer here, which we use to close this official session. How are we going to proceed? First of all, there will be a reception in the inner court of our university. And I would like to ask our guests already here, they already can go to the inner court and enjoy some coffee, tea, beer, wine, whatever is suitable over there. We will make a picture here together with the online members here. And I would like to ask anybody who likes to make a picture, remain here. Go to seat line 2, because that's the best line, and we will proceed. Thank you very much. Yep. You take the parliament. And we go stand.